Paulina,
I'm Sanvi.
and this is our Flight Lab report.
So, to start off our experiment, in the beginning of the semester, we were given two unknown mutants of Drosophila melanogaster. So, if we're looking at this figure right here, going from left to right, A was wild type, and it had dark red eyes. B was our first unknown mutant, and it was bug, and it had brownish red eyes. And then C was our second and last unknown mutant. It was called cog, and it had white eyes.
Then throughout the semester, we based our entire Flight Lab procedure on the following learning goals. And they are differentiate X linkage versus autosomal linkage in our unknown mutations, perform a three-factor mapping on our unknowns to determine the location on the gene, and develop a hypothesis about the gene and test it using a complementation analysis.
So the first cross we worked on was with wild type and bug. So for the male wild type flies, it wasn't that important if they were virgins or not because of the fact that they are prolific breeders. So we just lightly etherized some flies and collected as many males as we could. On the other hand, the mutated females had to be virgins since we needed to remain certain of the origins of the mutation. Because if they had been inseminated by the bug males, um, then that would have messed up our results and introduced ambiguity into the system, which is not what we needed. So in order to ensure that they were virgins, we cleared the adult flies early in the morning from the vial and then waited for 4 to 6 hours for new flies to close. Um, when we were collecting the virgin flies, we had to make sure that they were females. So we lightly etherized them and closely looked for sex combs because that would prove that they were males, and that's not what we needed. After we collected the proper amount of flies, we crossed 8 wild type males and 8 bug females. And we waited for around 10 days so that they could properly cross and have the amount of pupae that we needed. And the point of doing this cross was to determine the inheritance of the mutation. So after those 10 days had passed, we observed the sex and the eye color to determine the inheritance of this mutation. And then the exact same procedure was followed for the cross with wild type and cog. Um, and going to our results in conclusion, based on our F1 phenotypes, we concluded that the X-linked mutation was in cog and then the autosomal mutation was in bug. But to be a little more specific, we found that the cog mutation was X-linked recessive because if we take a look in our table right over here, in the cog wild type cross, all the males had the same eye color as the mother, which was white. But the daughter still had the wild type dark red eyes, which showed that all the affected mothers had caused their sons to be affected. Um, the bug wild type cross, on the other hand, was an autosomal mutation because now if we look into this column right here, we saw that all the offspring of the F1 generation still inherited the wild type eyes regardless of their sex. And this was just like the total number of all our flies that we got. So the total for the cog wild type was 110, and then the total for the bug wild type was 125.
Okay, to go off of that, the next part was three-point mapping. So in our three-point cross, we had to determine which one we would like to continue, and we went with cog due to the fact that it's an X-linked mutation. So if we were to do the three-point mapping, first we would have to establish two genes to cross with the mutant unknown, and that in this case it's Y and F. And then in the since the crossing over does not occur in Drosophila males in the X linkage, due to the fact that they only have one chromosome compared to the two of the female, when we were setting up the cross for the F1 generation, it would be important to use females instead of males. After the selected genes are tested, we would cross a wild type male of the unknown gene with a female who is mutant for Y and F. Next, we would have to cross the F1s with each other to get the F2 generation. And then after that, we would have seen a whole bunch of F2 generation and collect a sample and examine each phenotype and determine which combinations and traits occur most frequently in each other. And then here we have our simulated data that Dr. Elliott provided. So in order to calculate the recombination frequency between each of the pairs, we had to divide the number of recombinant offspring by the total number of offspring. The smaller the recombination frequency, the closer the genes are on the chromosome. So here we had to figure out the double crossover, the single crossover, and the no crossover. After getting these numbers on this chart, we determined that the 51.5% was the non-crossover, the 47.5 and the 1% was a single crossover, and the 0% would be our double crossover due to the fact that it involves two separate recombinations, making it least likely to occur.
So, using the simulated data from above, we calculated our regions and then constructed a genetic map which illustrated the distances between Y, U, and F. So, in order to calculate these regions, we compared the percentages of the single crossovers, which is what Paulina was just talking about. And we found out that Y and U were 1 mu away, suggesting that they would have a higher inheritance frequency because they had less chances of double crossover occurring. And then on the other hand, U and F were 47.5 mu away, showing that double crossovers would be more often and it would have a lower inheritance frequency. So based off of our genetic map and our information that we gathered from the whole semester, we hypothesized that our unknown mutant cog was on the same gene as the white eyes mutation W and the X chromosome. And since we saw that Y and U were 1 mu away in the last slide, we would need to find the eye color mutation that was the closest to Y in the chromosome. So if we look at this picture right here provided by Flybase, if you look at the X chromosome, which is this one, we saw that Y was yellow body and it was all the way at the top at 0.0. And we looked for the closest mutation, which was in the eyes, which was 1.5 and white eyes. So since this mutation was the closest to U from the genetic map, we could identify that white eyes is the one that we would use. And if our hypothesis was right, when we crossed cog and white with each other, we would see that the F1 offspring would have the mutant eyes.
So to test our hypothesis, we crossed cog virgin females with white males because in order to complete a complementation test, we want to determine whether two different mutations affect the same gene or different genes. When you mate a virgin mutant female with a male suspected to be carrying the same X-linked mutation, the F1 generation would not be expected to see the mutant phenotype, showing complementation not occurring. This is exactly what we saw in our cross. After crossing the cog females with white males, the offspring showcased the mutant phenotype. In conclusion, since the F1 generation showed the mutant phenotype, our test led to the discovery that the mutations affected the same gene, indicating that complementation didn't occur, meaning no wild-type eyes were seen. For future directions and other things we want to learn are the expression pattern of our mutation, interactions with other genes, and phenotypic variability. Next, to study our mutation further, we could possibly perform a whole genome sequencing, which is a comprehensive technique that sequences an organism's entire genome to identify genetic variants, including the mutations. WGS offers a comprehensive approach to finding mutations and analyzing genetic variation. It is a valuable tool for researchers seeking to understand the genetic basis of phenotypes and advance the field of genomics.
And these were just our references. Thank you so much.